Hi, my name is Naina, and I've been mentoring with Magic for about 2 years now. This year, I've been mentoring Lupita P. She is a 11th grader at Oxford Day Academy. Working with her has been such a pleasure. She is so talented and so bright. I just really enjoyed working with her. For her Magic project, she picked to create an animation about a gene editing technology called CRISPR, with the goal of learning more about it ourselves as well as educating others about it. She picked this project because she's interested in video editing as well as gene editing. So we thought, how can we mix both of these together to create a project? She is a super fast learner. She picked up most of the animation skills in a span of a few weeks. Overall, it was very easy to communicate and work with her since most of her meetings were remote, but we were able to work around it, and it was just a pleasure working with her. Without much further ado, I'm going to introduce Lupita now.
Okay, so now I'm going to introduce my mentor. My mentor's name is Naina. She is currently working as a computational biologist, which has nothing to do with my project. Originally, she didn't know, Ira didn't know that I was going to be working with DNA or an animation about it. So I think I got so lucky on that part. She is from India originally, and she moved to the US after her undergrad. She's currently working at a company called DNA Nexus. The company helps people analyze their genes. It works mostly with hospitals to help with the diagnosis of a patient. And on her free time, she enjoys drawing and sketching to keep inside with her creative side. Is this okay? So as Nana has said, my project has to do with an animation on gene editing method called CRISPR-Cas9. CRISPR-Cas9 is a molecular pair of scissors that allows us to edit the genome. With this technology, it would be possible to get rid of a mutation in the DNA sequence. An easier way to think about this is it was currently the allergy season. Theoretically speaking, with CRISPR, you can get rid of someone's allergies. I chose this project because of my interest in gene editing and video post-processing. The animation app that I use was Flip-a-Clip. It's compatible with iOS and Android. With this app, I learned how to copy and paste the drawings I made, which made my life way, way easier, since instead of having to draw 20 of the same drawings over and over and over again, I could just copy the same drawings 20 times. This helped with moving the objects look a lot more seamless. I learned the importance of frame per second. The amount of frames or individual drawings you see per second affects how fast or how slow an animation goes. For my research, I use Google Scholar to help with the accuracy and all the information that I would get with CRISPR. But since it's a little bit dense to read only articles on this topic, I also watched YouTube videos. The YouTube videos, I also did research for my animation video to see the different animation styles, design styles, and different explanation styles that already existed towards CRISPR and implement that into my own video. The outline for my animation is first talking about how CRISPR works, why is it such a big deal, and the ethical issues in regards to CRISPR. Because of the current pandemic, my mentor and I decided it'd be interesting to see what CRISPR is currently doing with COVID-19. Now, I'm going to be going over my animation video. I will be explaining it live, so please be a little patient with me if the animation isn't in sync with what I'm explaining. What does CRISPR-Cas9 stand for? CRISPR-Cas9 stands for clustered regularly short prodromic repeats. Cas9 refers to the Cas9 protein. What is CRISPR made up of? CRISPR-Cas9 consists of the target DNA and as stated in the name, the Cas9 protein. Along with the Cas9 protein, there's a guide gRNA. And this part is where you hear the boom, boom explosive noises that aren't here. The two main elements to CRISPR are, that was the two main elements of CRISPR. Cas9, with the help of gRNA, acts like molecular scissors. The gRNA helps guide the Cas9 protein to a specific part of the genome, allowing it to cut part of the DNA and even inserting a new sequence. Origin of CRISPR. It all started with research on the immune system that led to scientists taking a closer look at the Cas9 protein and how the protein combats viral DNA. The Cas9 protein chops up the invading genetic material. This is a natural occurring process that bacteria uses as a defense mechanism against viruses. This led to a very big discovery on the relationship between Cas9 and gRNA. When programmed correctly, it creates this powerful and precise molecular scissors. Why is CRISPR so important? The scientists discovered this thing. Why is it so important? What's the big deal? CRISPR allows geneticists and medical researchers to edit parts of the genome. Theoretically speaking, it would be possible to fix mutations in the DNA that cause diseases like cancer. Currently, CRISPR is being used to detect the presence of COVID-19 by a biochemist who co-founded CRISPR, Dr. Jennifer Doudna, and her colleagues at UC Berkeley. Their goal is currently to use their labs to test up to 2,000 samples per day. Ethical issues. One of the big ethical issues with CRISPR is the fact that it works better and is more effective and precise on a human embryo. There's concerns about whether or not it's morally right to mess with the DNA of an embryo. Another one of the ethical concerns with CRISPR has to do with religious concerns of humans playing with the role of God by messing with human DNA and whether it's right to edit the human DNA. The last concern has to do with when CRISPR becomes more developed and is potentially used for beauty purposes by parents to edit the traits their children will have. What happens when the child finds out that their DNA was messed with when they were merely embryos to enhance their looks? This brings out a bigger issue of consent. Those are the only ethical issues I'm going to be going over. This is the part where I would thank the viewer for watching. I hope this increased your curiosity towards CRISPR. There is an existing video with audio already in it, but for this presentation, I did have to explain it live. So I apologize for any mistakes I had with the syncing of my explanation and the video. For my magical experience, meeting my mentor for the first time in person was one of the most memorable moments. I remember being nervous and excited to meet her for the first time, especially since our first meeting was virtually and I had a few technical problems. One of the things that made this experience magical was the small, big milestones. Figuring out how to maneuver, flip a clip, the first little animation that was made, that was very exciting and meant that I could actually continue with this project and it was going to work out fine. The amount of frames in this animation is 2,207, which is a pretty big number. The challenges I had with this animation was the part where the Cas9 protein is supposed to punch the viral DNA. I was so focused on trying to get it to be like anatomically correct and stuff, but I thought, what am I doing? It's a Cas9 protein. It's hitting a viral DNA. It's fine. Exporting the animation was also one of the extremely hard challenges. I had to edit it for transitioning purposes, and since there's already a video out there that has my voice explanation in it, I had to use iMovie, but getting it to iMovie took so long, but I was able to do it in the end. Thank you.